There's a few things to remember about DNA before we go into how it replicates itself. One, DNA replication is semi-conservative with one template strand or parent strand and one daughter strand. DNA replication occurs during the S phase of interphase, be it mitosis or meiosis. And if you remember, S phase takes about 8 hours of a 24-hour period, whereas mitosis itself is only about 1. So this is not exactly a very fast process. Also, I hope you remember that the DNA nucleotide is numbered from 1 to 5 based off of the carbons. And the 3' end is where the OH group is, and the 5' end is where the phosphate group is. So DNA will always grow from 5 to 3. The process of DNA replication ensures that every new cell that results from a division has identical DNA. Enzymes facilitate the replication process, and we're going to learn about them as we go. But overall, you have a parent strand that splits in the middle and forms 2 new strands. The nucleotides themselves are bonded together with phosphodiester bonds, but bonds take energy, and that energy has to come from somewhere. The energy of replication comes from the nucleotides themselves, because they don't actually arrive as what we know as a nucleotide. They arrive as nucleosides. The example that you see here is one you should be used to. It's a ATP, essentially, even though it's a DNA form, because it has a deoxyribose sugar. So the real reason that nucleotides can be added to the 3' end is that, well, the energy is on the 5, and the OH group for dehydration synthesis is on the 3' end. So when you actually model it out, ATP is going to become ADP, and that's going to release some energy, and it's going to become AMP, and that'll release a little more energy, and that's what bonds in. Now that can be TTP, that can be GTP, that can be CTP, whatever. It's how they all come in. It's not just adenine that does this. So when they're actually coming in, the bonds form because an enzyme uses the energy from breaking those phosphate groups to make new connections. They attach across the middle with hydrogen bonding, but the phosphodiester bonding requires energy to build. And that is going to be the real reason why DNA is 5 to 3 when it grows, and that's also going to be the real reason why DNA is anti-parallel, because they have to connect across the middle. So one strand will be 5 to 3, the other strand will be 3 to 5. It'll always grow from 5 to 3, but the actual directionality within the structure will be different. In eukaryotes, DNA replication is initiated in multiple sites along a chromosome called replicons. Thousands of replicons, each covering 50 to 300 kilobases, form replication bubbles that fuse to, in the end, make 2 daughter double-stranded DNA molecules. A number of proteins and enzymes are responsible for this. It's a bit different in prokaryotes because their DNA is circular. They only have one replication bubble to form. Some of the enzymes are a little different, but they still have the same jobs. They might be structured a little bit differently, and there's a few other differences that we'll talk about at the end. To really understand DNA replication, you need to understand the enzymes that are involved. And while there is an initiator protein that makes the initial opening for the replicon, it's not super important. So we're going to start with helicase. So DNA helicase stimulates the separation of the 2 strands, and it's going to unwind the DNA at the origin of replication. Now that origin of replication is always going to be somewhere that's rich in adenine-thymine bonds because they only make 2 hydrogen bonds and it's easier to break. Topoisomerase is going to be the sort of swiveling around and untwisting and rejoining the strands, preventing your DNA from knotting up and breaking during this copying process. And the SSBs, or the single-stranded binding proteins, are going to bind to these single-stranded sections of DNA to prevent the reconnection. We don't want these things to come back together right now. We want to build a new second strand here, a new double-stranded molecule. And if they bound back together now, that would never happen. Once the opening is made and space is available, primase, RNA primase, is going to come in and it's going to make a small section of RNA primer. That primer will serve as a starting place for DNA polymerase III, which is our main DNA builder. Now, DNA polymerase III is very limited. It can only grow in 5 to 3. It can only attach to a double-stranded structure. So the RNA primer makes a double-stranded section for it to attach to so that it can copy new bases on to the parent strand. Now, it's very, very fast. It's about 1,000 bases per second, but just like when you do fast work, it tends to be inaccurate. So there tends to be lots of errors. DNA polymerase III is our main DNA builder, and it builds on both strands. But we need to realize, since DNA is anti-parallel and polymerase can only grow from 5 to 3, the strands are going to be different. So my parent strand that was originally 3 to 5 will be making a new strand that is 5 to 3. So DNA polymerase is not going to have any problems with that. It's going to be a continuous strand, and it's going to go a little faster, so we call it the leading strand. The other strand, since originally it was 5 to 3, the new strand that's being made is trying to be 3 to 5, and that's not a simple process, and it's not going to happen. So what happens is we use RNA primase to continually make new primers on the lagging strand, the opposite strand. That is going to allow polymerase to have enough grip, basically, to work against the flow for small stretches of time. It's going to make these discontinuous sections of DNA that we call Okazaki fragments. So those Okazaki fragments can be seen here. Small stretches of 5 to 3 DNA that is growing against the flow of my other enzymes. So while replication wants to go towards the replication fork, it can't on this parent strand because it's already 5 to 3 and the new one is 3 to 5. So the leading and the lagging strands are, like we said, are based off of the differences in what we see. If you look at that as a structure, the leading strand is making a 5 to 3 molecule, and it has no problems with that. Now we know, of course, that enzymes are used breaking energy and so on, so the leading strand works like that. The lagging strand, however, is making a 3 to 5 strand. So that's not going to work because I don't have an OH group to bond to. So what happens is you make small sections of 5 to 3, and you use a new enzyme called ligase to seal those strands together. It's basically going to be the DNA glue that's going to form phosphodiester bonds wherever they couldn't form on their own. Once ligase and DNA polymerase III have done their jobs, a new polymerase comes in, DNA polymerase I, and it's going to remove the RNA primers. Now, DNA polymerase I is called DNA polymerase I because it was the first enzyme discovered with polymerase activity, and it is really the most well-known one. It is not, however, the main DNA builder, but it is extremely important. It replaces the RNA primers and turns them back into DNA, so you don't have RNA sections, which is good. It also fixes the mistakes, or most of the mistakes, that polymerase III makes. It is a lot slower enzyme, but it's able to take your mutation rate from about 1 in 10,000 to about 1 in 100 million. Does that mean that you don't still have mutations? Of course not. Of course you do. But DNA polymerase I is going to be able to fix things like thymine dimers, which happens when you get a sunburn. Well, nuclease, polymerase I, is going to come in and it's going to cut out the damaged section, build a new section, and ligase will seal the gap. So this is how you fix mutations most of the time. The ends of DNA turn out to be a very particular problem, and we call those telomeres, which you can see here. DNA polymerase I cannot replace the final primer on both strands. It's still a polymerase. It still needs to be able to build on a double-stranded structure, and it can't do that at the end. So what happens is that, well, that primer leaves a one-stranded section. That section becomes weak, it falls off, and your chromosomes get shorter. And this is going to happen every time your chromosomes copy. So what we see is that the telomeres turn out to be expendable DNA, lots of junk. For humans, it's TTA, GGG over and over and over and over and over again. Eventually, after enough copying, the telomeres will be lost, and that leads to cellular aging. Now, there are certain enzymes that can fix this, but the only times we really see that in humans are in cancer, and that is what leads to the functional immortality of cancer cells, because they're constantly able to repair their telomeres. This is a more detailed summary of what happens at each level of DNA replication. If you are still confused by something, I would suggest looking over this. If you have any more questions, let me know and I'll be happy to help. Outside of the differences that we've talked about already, there are a few other differences in prokaryotic and eukaryotic replication. Obviously, prokaryotic replication happens in the cytoplasm because they don't have a nucleus. Other differences that we didn't see so far is that while prokaryotic Okazaki fragments are very, very large, eukaryotic ones are very, very small. We don't know if that has to do with complexity issue or enzyme issues. Not 100% certain. The replication for prokaryotes is much, much faster because they do no proofreading. There is no checking for mutations. That can be very, very bad. Basically, diseases can accumulate, or it can be very, very good because mutations that turn out to be beneficial accumulate and the prokaryotes are able to evolve some new feature. So it's a give and take balance here.